Good morning, everybody. Do you hear me? Yes. Good morning, everybody. Come on in the class. Do fast. Let's start with the metabolism of nucleotides, that is purine. Purine metabolism. Am I audible? Okay. Yes. Today's topic is metabolism of nucleotides, that is metabolism of purine. What is purine? Purine is a nitrogen base like adenine and guanine. It includes adenine and guanine. This is the structure representing the sources of purine ring or the sources which creates a purine ring, that is nucleotides originally. See here, this N1 comes from aspartate amino acid and 7C5C4 comes from glycine amino acids. Likewise, C8 and C2 comes from N10 formyl tetrahydrofolate and amide nitrogen of glutamine gives N3 and N4 nitrogen to purine ring. Lastly, respiratory CO2, that is CO2, gives carbon number 6 to the purine ring. Am I audible? Okay. So this is a structure of purine ring, aspartate, glycine and glutamine. 3 amino acids contribute in the formation of purine ring. First, we will discuss about the synthesis of purine. Purine is a nitrogen base, as I told earlier. This is only present in the way of, in the structure of, in the form of nucleotides, not lonely present like nitrogen base. First, we will discuss the synthesis of purines. There are two methods of synthesis of, or we can say biosynthesis of purines. First is de novo synthesis and other is salvage pathway. First, we will discuss de novo synthesis of purines. The basic compound or basic structure which we require is ribose 5-phosphate, which we require is alpha D ribose 5-phosphate. What is this ribose 5-phosphate? Ribose 5-phosphate is a pentose sugar, that is a nucleotide which is a mixture of sugar and phosphate. This ribose 5-phosphate in the presence of PRPP synthetase. What is this PRPP? PRPP is 5-phosphoribosyl alpha pyrophosphates. What is pyrophosphates? POP. That is, pyrophosphates are diphosphates. Basically, they are diphosphates. So, in the presence of ATP, alpha D ribose 5-phosphate converts into 5-phosphoribosyl alpha pyrophosphate, that is PRPP. ATP converts into AMP. This is the first step in the process of de novo synthesis of purines. This is the whole pathway or whole process of the synthesis of purine nucleotide, or we can say de novo synthesis of purine nucleotides. Ultimately, we get the IMP molecule or purine, that is inosinate IMP, inosinate monophosphate. We will discuss one by one. Yes. First, we can say we have PRPP, 5-phosphoribosyl pyrophosphate, that in the presence of amino acid glutamine, it converts into 5-phospho-beta-ribosylamine. That is, glutamine converts into glutamate in the presence of glutamine PRPP amidotransferase. That is the most important enzyme or regulatory enzyme of in the whole pathway of de novo synthesis of purine. We can say here this 5-phospho-beta, first it is in alpha position, H is above the ring, and in the 5-phospho-beta ribosylamine, H present in the below position. So it is called as beta D ribosylamine. Then, am I audible? Okay, please reply me. Then, in the presence of, that is GAR synthetase, in the presence of GAR synthetase, we can get glycinamide ribonucleotide, that is in the presence of ATP. ATP converts into ADP and inorganic phosphate or inorganic phosphorus. Then glycinamide ribonucleotide, that is GAR, in the presence of GAR transformylase, in the presence of N10 formyl H4 folate. Here it is important, the coenzyme which is used in this process is folic acid. Without folic acid, this do not occur. The whole process is very important because it requires the coenzyme which is vitamin B complex, that is folic acid or folate. In the presence of this N10 formyl H4 folate, it is converted into formyl glycinamide ribonucleotide. We can say this here NH2, then amide, CONH, glycinamide, and then formyl from N10 formyl H4 folate. Next, this formyl glycinamide ribonucleotide again combines with glutamine, and in the presence of FGAR amidotransferase. Enzyme represent that amidotransferase, transfer of amide group. FGAR, that is formyl glycinamide ribonucleotide. In the presence of ATP, it converts into ADP and pyrophosphate at inorganic phosphate and converted into formyl glycinamide ribonucleotide. You can see NH, NH here. Then again, this is FGAM cyclase, or we can say another name of FGAM cyclase is AIR synthetase. What is this AIR synthetase? It is a very important molecule in the whole process. That is 5-amido-imidazole ribonucleotide. 5-amino-imidazole ribonucleotide, that is AIR. AIR means A for amino, I for imidazole, and R for ribonucleotide. This is important. R represent ribose 5-phosphates. Please listen it carefully. R represent ribose 5-phosphate. So don't confuse what is R. R is nothing. R represents ribose 5-phosphate. Here we can get AIR. Yes, from AIR, that is 5-amido imidazole ribonucleotide, we can say this is the enzyme N5-CAIR synthetase. That means, again, we can see here the enzyme is AIR carboxylase. It means incorporation of CO2. That gives the carbon in the position of 6, C6. First, in the first step, we can say the enzyme N5-CAIR synthetase converts into AIR into N5-CAIR. Then in the presence of mutase or isomerase, we can see the change in the position and we can get carboxyamino-imidazole ribonucleotide, that is CAIR, carboxy-amino-imidazole ribonucleotide. Carboxy-amino-imidazole ribonucleotide, CAIR. Then, in the presence of that synthetase enzyme, here aspartate incorporates and ATP is essential for this process. Then ATP converts into ADP plus inorganic phosphorus. We can get N-succinyl 5-amido imidazole 4-carboxamide. Here carboxamide means CO2 incorporates or already incorporated ribonucleotide, that is SAICAR. It means this whole process combines with aspartate. In the last phase of the synthesis of purine nitrogen base, we can see that is SAICAR lyase. What is lyase? Lyase is the enzyme which removes the molecule, like any molecule by this enzyme. N-succinyl 5-amino imidazole 4-carboxamide ribonucleotide, that is SAICAR. In the presence of SAICAR lyase, removes fumarate, and we can find we get 5-amino imidazole 4-carboxamide ribonucleotide, that is AICAR. Here we can see the enzyme AICAR transformylase. In the presence of again coenzyme N10 formyl H4 THF folate converts into H4 folate, and we can get N-formyl amino imidazole 4-carboxamide ribonucleotide that ultimately converts into IMP. Our emphasis is to get inosinate, that is IMP. What is inosinate and inosinate? IMP is a nucleotide which is made up of inosine, that is nucleoside, and the bioactive peptide, bioactives purines. Here we can see the closure of the ring again, and we can see that C2, carbon C2 in the purine ring comes from that N10 formyl H4 folate. Am I audible to all of you? Okay, please write. This whole process ultimately emphasizes on the synthesis of inosinate. What is this nitrogen bases? First, we can discuss again. This N2 bases are purines, adenine and guanine. Then nucleoside. What is nucleoside? Nucleoside is a mixture of sugar and nitrogen base. Sugar is ribose 5-phosphate. You all know. Then we talk about nucleotide. What is nucleotide? Nucleotide is, what is nucleotide? Yes, please write. What is nucleotide? What is nucleotide? Please write. What is nucleotide? Nucleotide is made up of, please write down. Yes, what is nucleotide? Nucleotide is made up of nucleoside plus phosphate, or we can say, yes, good. Tamanna, yes, Tamanna, am I audible to you? Yes. This IMP, yes, I am talking about nucleotides. What are those nucleotides? Nucleotides are made up of nucleoside plus phosphate, or we can say sugar, phosphate, and nitrogen base. First, we will talk about adenine. The purine nitrogen base is adenine. From adenine, we can get adenosine. Adenosine is a nucleoside, and then AMP. AMP is adenosine monophosphate. That is adenosine monophosphate. It is a AMP. It is a name of nucleotide, which is made up of adenine plus phosphate plus ribose 5 phosphate plus ribose sugar. That is combined. We can say ribose 5 phosphate. Then I think it is clear to all of you what is AMP. Yes. Now we go further. We found a molecule that is IMP inosinate. It is an important molecule. So by de novo synthesis, we can get the molecule IMP or inosinate. IMP is again a nucleotide. AMP is also called as adenylate. The whole process occurs in liver mainly and in the cytoplasm of the cell. Then comes into the process of biosynthesis of AMP and GMP from IMP. It is a very important process again because we should have AMP and GMP for all the metabolic processes. So we can see here this inosinate IMP, inosine monophosphate, in the presence of adenyl succinate synthetase. This is the important enzyme. Then incorporates aspartate, and the presence of GTP. GTP converts into GTP plus inorganic phosphorus, and it will give us adenylate succinate or adenyl succinate. This whole, we can say this ribose 5 phosphate, and this adenyl succinate. Then again removal of fumarate by the enzyme adenyl succinate lyase, and we can get this AMP. AMP, or I already told you what is AMP, adenosine monophosphate. Adenosine, I repeat, again it is adenosine monophosphate. Then we will discuss the formation of GMP from IMP. What is GMP? Again, I would like to clear you that nitrogen base guanine. There are two bases which is related to purine nucleotide or purines. First is adenine and second is guanine. Guanine is a nitrogen base and nucleoside is guanosine. And GMP, that is guanylate or guanosine 5 monophosphate, is a nucleotide of guanine. It is very important in the presence of IMP dehydrogenase, NAD, NADH. Here you can see dehydrogenase. Dehydrogenase means the process of oxidation. Then S2 combines, H2O combines, and we can get this genthylate or XMP. XMP is, what is XMP? It is also a bioactive purine, that is xanthine xanthosine monophosphate is XMP. Then we can see here XMP glutamine amidotransferase. In the presence of XMP glutamine amidotransferase, we can get GMP, that is, I already told you, guanosine 5 monophosphate, that is guanylate. In the presence of ATP, ATP converts into AMP and PPI. Again, H2O combines in the process. This whole process is very important because By this process, we can get AMP, which is a very valuable molecule for all the metabolic process. You all know cyclic AMP and AMP itself for the synthesis of nucleic acid and other nucleotides. Yes. Another pathway of purine synthesis is salvage pathway of purine synthesis. That is again very important and basic pathway of purine synthesis, and we can say biosynthesis of purine nucleotides. I already told you purine doesn't alone synthesized. It is synthesized in the form of nucleotides. This is the main important feature of this purine biosynthesis. First is de novo synthesis by the precursors of we can achieve the purine, and in the salvage pathway by already present molecules like nucleotides, nucleosides, and nitrogen bases. First, we can say nucleic acid. The feature of this whole cycle, like central dogma, we can see nucleic acid. The enzyme nuclease, ASE, it represents the hydrolysis of nucleic acid. You all know. Am I audible, Tamanna? Please write in the box. Yes, nucleic acid converts into nucleotides. Nucleotide in the presence of nuclease. ASE, it is the process of hydrolysis of nucleic acid. It is the process of hydrolysis of nucleic acid. Nucleic acid both combines first DNA and RNA. You all know about DNA and RNA. Then in the presence of nucleotidases and phosphatase, we can get nucleosides, because you all know that nucleosides is the first step. Then nucleosidase, in the presence of nucleosidase enzyme, the hydrolysis of nucleoside, we can get bases. And another two steps here. First is catabolism, degradation product, and other is mononucleotides. Again, it forms mononucleates and mononucleotides and polynucleotides, and other. Again, we can get nucleic acid like DNA and RNA. This is the basic pathway that is called as salvage pathway. It is present in almost all the cells. This is the main pathway of salvage process of purine synthesis, that adenine, it is converted into AMP. You all know adenosine monophosphate, that is a nucleotide. In the presence of adenine phosphoribosyl transferase. The enzyme is important, very important, adenine phosphoribosyl transferase. In the presence of PRPP and PP, it converts into PPI, pyrophosphate inorganic. What does it mean? That PRPP gives the structure of ribose 5-phosphate to all the nitrogen bases. So we can get the nucleotide in the combination of nitrogen base, that is adenine, ribose 5-phosphate, that is ribose, sugar, and phosphate itself. So we can get nucleotide adenosine monophosphate, that is called as AMP of or adenylate. Next is hypoxanthine. Hypoxanthine, in the presence of hypoxanthine guanine phosphoribosyl transferase. This is a very common enzyme that is called as HGPRT. That is called as HGPRT. In the presence of PRPP, again, it converts into PPI or inorganic pyrophosphate. It converts into IMP. What is IMP? Inosine monophosphate. Then guanine, again, in the presence of this HGPRT, hypoxanthine guanine phosphoribosyl transferase, we can get GMP. GMP is guanosine 5 monophosphate, that is guanylate. It is a nucleotide of guanine in the presence of PRPP, and again we can it converted into PPI or inorganic pyrophosphate in the presence of hypoxanthine guanine phosphoribosyl transferase. Yes, by structure we can say this slide also we can understand easily by this slide. This adenine, The enzyme is adenine phosphoribosyl transferase, PRP PPI. You can see AMP here. Incorporation of ribose 5 phosphate. You can see here ribose sugar, phosphate, and this phosphate and this whole adenine. This is called as adenosine monophosphate. In the next, we can say IMP, and we can see IMP and GMP by the enzyme HGPRT, that is hypoxanthine guanine phosphoribosyl transferase by hypoxanthine and guanine. This is the important process related to many metabolic reactions and the synthesis of AMP, because you know CMP, that is cyclic AMP, is the second messenger in all the metabolism. Another is GMP and IP. GMP and IP. Here, the purpose of PRPP incorporation is only the incorporation of ribose 5 phosphate, that makes a complete AMP or complete nucleotide, that is called as adenosine monophosphate, likewise IMP and GMP. Next, here we can see the NMP, that is called as nucleoside monophosphate in the presence of kinase. As I told you many times that the enzyme is kinase. Kinase is responsible for the phosphate transfer or phosphorus transfer. Here we can see ATP converts into ADP. The transfer of phosphate or phosphorus, that it creates nucleoside diphosphate, and in the presence of kinase enzyme, ATP converts into ADP and we can get nucleoside triphosphate. Here, one process is also important, that is the conversion of ribonucleotides to deoxyribonucleotides. What is DNA? Please write down the full form of DNA and RNA. Please write in the box the full form of DNA and RNA. Yes, what is deoxy? This DNA represents deoxy. On which carbon oxygen is absent? Which carbon? Yes, second one, C2, right. There you can see the ribonucleotide diphosphate converts into deoxyribonucleotides in the presence. It is important enzyme, name of enzyme is important, ribonucleotide reductase. It is a very important enzyme, ribonucleotide reductase. Here in the presence of NADP, NADH converts into NADP, and the main or basic coenzyme is, that is protein thyrodoxine. It is a very important protein. The whole process requires this thyrodoxine. It is a very important protein, or we can say coenzyme, that requires in the whole process. Yeah, this represent oxidation, and here you can say oxygen lacks. You can say oxygen is not present here, and in this structure oxygen is present. Am I audible to all of you? Yes? Okay. Next one is purine catabolism. Till now we have studied the synthesis of purine. Till now we have studied the synthesis of purine. Two processes are there. First, de novo synthesis, and another is salvage. The process of de novo biosynthesis of purine, it incorporates the precursors, and another is the salvage pathway, which is formed, or which is Purine is synthesized by the basic nucleotides, nucleosides, and nitrogen bases, which are already present by the catabolism of purines. This slide show the catabolism. Metabolism involves two processes. First is the synthesis. First is the synthesis of purine, that is called as anabolism, and another process is catabolism, that is degradation of purine, which is also equally important. This is the process of catabolism or degradation of purines. We can say this adenosine. Adenosine, what is adenosine? Adenosine is a nucleoside. Adenosine is a nucleoside, and this adenosine deaminase, the presence of adenosine deaminase means this enzyme is very important. I will tell you further why it is so important, because this enzyme is involved in gene therapy first time. So this enzyme is very important. It is called as adenosine deaminase. Deaminase means deamine. Deamine means removal of amino group. You can say here. You can clearly see here NS2. Here there is no NS2, and then we can find or we can get inosine. Please check, this is a molecule of inosine, and then this inosine, you can say here, it converts into hypoxanthine, and again hypoxanthine converts into xanthine. The basic and the last component or compound which is found from the catabolism is uric acid, which is a very important. The molecule is uric acid. It is although a bioactive purine. It is although a bioactive purine. In the presence of this enzyme xanthine oxidase, there are two enzymes which are very important. First is adenosine deaminase, and another is xanthine oxidase. So whole process indicates the production or ultimately synthesis of uric acid, which is a desirable or desirable molecule, or which is the ultimate product of purine catabolism. You have to learn this adenosine deaminase, and then removal of ribose 1-phosphate, likewise by guanosine, and then creates both inosine and guanosine creates xanthine, and xanthine, in the presence of xanthine oxidase, we can get uric acid. This is the ultimate product of purine catabolism. Next, this slide shows clearly the whole purine catabolism. There's GMP, guanosine here, guanosine deaminase, likewise adenosine deaminase. You can say this nucleosidase. We can get hypoxanthine. From hypoxanthine, we can get xanthine oxidase. In the presence of xanthine oxidase, we can first get xanthine and then uric acid, the ultimate product of purine catabolism, that is uric acid. This is adenosine, this is hypoxanthine, this is xanthine, and this is uric acid. This is guanosine, that is GMP, because first is we require GMP and AMP, that is nucleotide. Nucleotide converts into nucleoside, that is adenosine. Then adenosine again converts into other biological active purines like hypoxanthine and xanthine, and then we can get the ultimate product, that is uric acid, by the purine catabolism, by the process of purine catabolism. It is a degradation of purines. We can see here GMP and AMP are the nucleotides, guanosine and adenosines are nucleosides, and then again it converts into other biologically active purines, and then ultimately it converts into uric acid by the enzyme xanthine oxidase. Here in the primates, birds, reptiles, and insects, this is in the form of uric acid. Uric acid is excreted. You all know. Another is allantoin in most mammals, bony fishes, allantoinate, and urea in the amphibians, marine invertebrates, fishes, and cartilaginous fishes, etc. So the main product of purine catabolism is, which is excreted. It is called as uric acid, mainly present in primates, birds, reptiles, and insects. These are the enzymes: urate oxidase, allantoinase, allantocase, and last one is urease. You all know urea converts into ammonia, which is water soluble. This is the whole pathway of purine synthesis and catabolism. This is a very important slide. You can clearly understand the whole process of salvage biosynthesis of purine and also the process of catabolism of purine. Am I audible? Again, please write. Am I audible to all of you? Okay. This is the whole process of purine synthesis and the catabolism. I repeat again, this is the whole overall salvage pathway and the catabolism. Overall salvage pathway of purine synthesis and the catabolism of purine also. What is this APRT? This is the adenosine phosphoribosyl transferase. You all see here, it is a adenine. Please, here this is a adenine. It is a nitrogen base, you all know. Next is in the combination of PRPP, adenine plus PRPP, in the presence of APRT, we can get AMP. We have already studied, isn't it? Yes. Then AMP again degrades or catabolized into adenosine in the presence of nucleotidase, and again in the presence of nucleosidase, it converts into adenine. This is the whole cycle of the production of purine and the catabolism of purine. We can get adenine, and by this adenine and PRPP incorporates ribose 5-phosphate, as I told earlier, and it creates or it produces the molecule of AMP, that is adenosine monophosphate. It is a nucleotide. Nucleotide, in the presence of nucleotidase, it converts into nucleoside, that is adenosine, and again adenosine, that is in the presence of nucleosidase, it converts into adenine. Okay, now it is clear to all of you what is the cycle. Please write down. Nucleotide converts into nucleosides, and nucleosides convert into nitrogen bases, and again nitrogen base adenine combines with PRPP in the presence of APRT, and again it forms AMP. Please write down in the box how AMP degrades and forms. Nucleotide converts into nucleosides, and nucleosides converts into nitrogen bases. Please write down, any of you, what is the whole process? Nucleotides, how nucleotide converts into a nitrogen base and how nitrogen base converts into a nucleotides. Please. Have you understood? Please write down in the box. Yes. Yes. So we will go further. Okay. Next is, here you can see this adenosine converts into inosine in the presence of adenosine deaminase, which we have studied earlier. Okay, adenosine deaminase. This is the important enzyme, as I told earlier. It is used first time in the process of gene therapy. This IMP, you can say here, IMP, this is the nucleotide. This XMP, it is also a nucleotide. GMP, it is also a nucleotide. Likewise, they are converted into inosine, that is a nucleoside, xanthosine nucleoside, guanosine nucleotide. Now it is clear now? Yes. XMP, GMP. These all are nucleotides. Inosine, xanthosine, guanosine. These are nucleosides. Then the enzyme. This nucleosidase converts into hypoxanthine. Again, we have studied xanthine oxidase convert into xanthine, and this guanine, in the presence of guanine deaminase, we can get xanthine. Now, this guanine plus PRPP converts into GMP. We have already studied this whole process. The important one is, again, this hypoxanthine in the presence of PRPP is this process of salvage process of biosynthesis. In the presence of HGPRT converts into IMP. And it is same here, guanine plus PRPP in the presence of HGPRT, we can get GMP. We can get GMP. This is a nucleotide, guanosine 5 monophosphate, or we can call it guanylate. I think it is clear to you all. This is the whole overall salvage pathway of purine synthesis and catabolism. The basic fundamental thing is nucleotide converts into nucleosides, and nucleoside converts into nitrogen base, and again nitrogen bases combines with ribose 5-phosphate through PRPP, and again it forms nucleotide. Do you understand this thing? Please write down yes or no. Yes. Regulatory mechanism in the biosynthesis of adenine and guanine nucleotides in E. coli. You can see here the feedback mechanism like hormones and enzymes. We can see if ADP, AMP, GMP, IMP are available in sufficient quantity or in more quantity, it inhibits the further synthesis of these 5-phosphoribosyl lamine or the synthesis of purines. IMP, AMP, and then IMP, it means IMP, then GMP and AMP. AXMP and JMP, all of the purines. If ADP, AMP, GMP, IMP are in sufficient amount or in more quantity by feedback mechanism, it regulates the whole process, because you can see here ribose 5-phosphate converts into PRPP, and there's a regulatory enzyme. Which one is? That is PRPP glutamine amidotransferase regulatory enzyme, the crux of the whole mechanism. This regulatory enzyme is a very important enzyme. This process or AMP, GMP, IMP inhibits feedback by feedback mechanism by the to the all purine biosynthesis process. This whole purine biosynthesis process is very important because of many nucleotides, many metabolism. It requires, I told earlier, like AMP, GMP, CMP, and any other. And the basic or main molecule is nucleic acid, like DNA and RNA. The last thing which I want to discuss to you all, that is disorder of purine metabolism. The very important is gout. It is a disorder associated with overproduction of uric acid, because the end product of purine metabolism, the ultimate or end product of purine metabolism of purine catabolism is, please write down, the end product of purine catabolism is, please write down, what is the end product of purine catabolism? Yes, uric acid. And yes, please write down in the box, the end product of purine catabolism, the whole process of catabolism of purine nitrogen base or nucleotides. Yes. There are two types of gout. First is primary gout and another is secondary gout. It is an inborn error of metabolism due to overproduction of uric acid, because it is most related to increased synthesis of purine nucleotide because of the overactivity or because of the increased activity in PRPP or PRPP enzymes or the PRPP synthetase, or by which we can get the increased synthesis of purine nucleotides. So when purine nucleotides are more, then catabolism more and the uric acid production is more. Secondary gout, secondary hyperuricemia, it is also called as hyperuricemia, because the in the blood we can find this uric acid. So it is called as Secondary hyperuricemia is due to various diseases, and this important is Lesch-Nyhan syndrome. It is caused by defect in the enzyme HGPRT. The patient of this disorder have an irresistible urge to bite their fingers, lips, often causing self-mutilation. It is a mental disorder type disease. And last one is SCID, disorder of purine metabolism. Is SCID, this is severe combined immunodeficiency in which both T cell and B cell dysfunctions. This is very important enzyme because adenosine deaminase, that is called as ADA, it involves in the ADA gene therapy, first attempt for gene therapy in modern medicine. So this enzyme is very important, adenosine deaminase, very important for your examination purpose because first attempt for gene therapy in modern medicine, transferring ADA gene in a girl suffering from SCID, that is called as severe combined immunodeficiency, SCID disease. And the last, we can see the diagrammatic picture of this disorder of purine metabolism. This is from Satyanarayan. You can also see from Satyanarayan this whole process. It's not clear slide. So now I repeat. The biosynthesis of purine and catabolism of purine. The whole metabolism, this purine anabolism or biosynthesis and catabolism includes metabolism of purine. So we have two processes. First is the de novo process, de novo synthesis of purines, mainly in liver, in the cytoplasm, in the hepatocytes of the cytoplasm of hepatocytes. First, the main precursor of the whole purine is ribose 5-phosphate. We can get by ribose 5-phosphate PRPP. And then the whole process, salvage pathway, you can see the salvage pathway. Nucleic acid, nucleotides, nucleoside, bases, and again we can get mononucleotides. This conversion of ribonucleotides to deoxyribonucleotide in the presence of ribonucleotide reductase. And then the whole purine catabolism, ultimately end product of this purine catabolism is uric acid, which is mainly found in primates. Now this topic ends, and any queries you can write in the box. And also I will take a test tomorrow on this whole purine metabolism. Am I audible to all of you again? Yes. How many people? Who can hear my voice? Have you understood the whole process of this purine metabolism? De novo process, you have to learn. There is no alternate except learning, and salvage is easy to all of you. You can easily understand. Our next topic will be related to pyrimidine catabolism or anabolism, that is pyrimidine metabolism. I will tell you later the time of the class. Tomorrow, a quiz will be taken from this whole topic, purine metabolism. You can get the PDF of this whole purine metabolism. You can get from this platform, Rajuva's. So you have to Learn 3 points: de novo, salvage, catabolism, that is uric acid. 3 main points: de novo synthesis from ribose 5-phosphate, PRPP forms, and by nucleosides, we can get nitrogen bases, and again from nitrogen bases we can get nucleotides. This is the salvage pathway. And then catabolism, the main process is the production of uric acid. The end product of whole purine metabolism is uric acid. Disorders like gout, Leishman, second is, first is gout, second SCID, and last one you can say, this last one is, you all know, the HGPRT deficiency syndrome. We can call it Leishman syndrome or HGPRT deficiency syndrome. So thank you very much for listening me. Tomorrow I will take a test of from this whole purine metabolism, and I will tell you the next class, about next class later on. Thank you once again. Mansi Sharma, Shashank Singh, Ankita, Amsuru.